Okay, good afternoon, everybody. Welcome to our monthly seminar, RadNet series. So I think I just want to briefly introduce our RadNet series and also RadNet in case people don't know about our RadNet initiative. So I think RadNet is being formed of the 4 institutes within sort of London Institute, UCL, King's College, Barts and Crick. And I think that this RadNet is initiated so that it can facilitate radiation-based research and it stimulates sort of collaboration and people sort of can collaborate with each other. And also we invite speakers who has been on the forefront of radiation-based research so that we can facilitate collaboration between us and the speakers and also to learn from them. So I think that there's a few things that I need to say before we actually introduce the speaker. So first of all, please mute your microphone and turn your camera off during the talk. And if you've got any questions or comments, you can either sort of post it in the chat, or you can raise your hand at the end of the talk. And then you can ask Professor Demaria directly, or I can ask on your behalf, whichever will be fine. And just to know that the meeting will be recorded. So I just want to introduce Professor Demaria. So we are very, very honored to have her giving us a talk today. So Professor Demaria originally from Italy, and she got her MD degree from University Turin, and then she went to do a postdoc in immunology in the US, specifically in New York City. And after that, she finished a residency in pathology at New York University School of Medicine. And following that, she became a professor at the Cornell, and she's well known for her study looking at synergy between radiation and immunotherapy. And specifically, she was the first person who showed that the radiation can convert the tumor that is not responsive to immunotherapy to become responsive to checkpoint inhibitor. And also she's in the forefront of investigating scopal effects due to radiotherapy. And she has a sort of special interest to see how radiation can induce sort of in situ vaccination effect in preclinical models and also in cancer patients. So we're very, very excited today that she's going to give us a talk about her work. And so we welcome her to give us a speech now. Thank you very much, Professor Demaria.
Thank you very much for the introduction and for the invitation. It's a pleasure. And I will now share my screen. Just a second, there we go. So we are good. Does it show well?
Yes, we can see very well. Thank you.
Okay, so this is just Cornell on the East River in Manhattan. These are my disclosures. None is directly relevant to today's talk. So I would like to first discuss what have been emerging as key mechanism of radiation induced in situ vaccination or pro-immunogenic effect of radiation. Then discuss what I think are important limitations to keep in mind, and conclude with discussing barriers defined here really as immunosuppressive mechanism that can be exacerbated or induced by radiation itself. So as you heard from Dr. Kong, I'm actually a pathologist by training, and I like to look at tissue. And the reason I'm showing here this picture, which is actually a primary triple negative breast cancer that was, from a patient. And I'm showing it to make the point that some patients really develop and retain meaningful spontaneous anti-tumor immune responses. This patient never was treated, just underwent surgery. You can see the tumor is full with infiltrated lymphocytes. Many of them are CD8 T cells, and there is a lot of induction of PGL1. This is true for many tumor types. There are patients that have this evidence of engaging the immune system. And this, has been very nicely reviewed here by Jérôme Gallon and colleagues, how this impacts prognosis and response to therapy. And actually, it's true not only for response to immunotherapy, but response also to other, let's say, conventional treatment. This is an example. We did analyze a large number of patients with triple-negative breast cancer. And the key message here is that, this patient underwent surgery and then AG1 chemotherapy, so very much standard of care. But those that had more infiltrated lymphocytes in their tumor have a better recurrence-free survival and overall survival. So it is quite important to think that, we should really find ways to change tumors that are, colloquially called as cold because they don't have infiltrated lymphocytes, such as the example shown here on the right, into tumors that are actually these cold tumors when it comes to immunotherapy. And I'm using here actually with this very nice cartoon from a review a few years ago by Panchal and Jimalis, and they were making the point that when immunotherapy works, it can induce these very long, durable responses. And so you want to find a way, but only in a small fraction of patients. And how do you extend this? How do you increase the curve to benefit more patients? And you have to find ways to convert a cold tumor into a tumor, because immune checkpoint inhibitors really work only if the patient has T cells against the tumor. And so can radiation be used for this goal? We believe, of course, it can, but we are still learning how to best do that. And before I go into the mechanism, I want to use this very cartoon from a very nice review of one of your colleagues in London, Caetano Reisi Souza, that summarizes the role of a very important subset of antigen-presenting cells known as conventional dendritic cells type 1. And what is the take-home message here is that these dendritic cells are really specialized in cross-presenting tumor antigen to CD8 T cells. And recent evidence from Bob Schreiber Lab suggests that they are also important for cross-presenting tumor antigen to CD4 T cells. But in any case, they are critical for development of anti-tumor immune responses, and they can take the antigen in the tumor, go to the lymph nodes when they prime anti-tumor CD8 T cells, but they also have a role in the tumor microenvironment. When the T cells that are primed in the draining lymph node come back, they really need to be reassured by this CDC1 that can, reactivate these T cells, produce cytokines that sustain their activity. And so the concept that is actually proposed in this review paper is that if we have a therapy that can increase the number and activation of CDC1 in a tumor, it might actually be able to increase patients' responses to immunotherapy. So one very important cytokine that can increase the recruitment and activation of CDC1 into the tumor is type 1 interferon. This was shown years ago by Tom Gajewski and Bob Schreiber's lab in mice models. So interferon type 1 is one of the factors that helps the recruitment and activation of this CDC1. They are also known as butterfree dependent dendritic cells because this transcription factor is key for their development. So they become activated, go to the lymph node, prime CD8 T cells that then can go back and eliminate the tumor. And so what we and others found is that radiation can trigger type 1 interferon production by in the tumor, and an important compartment when this occurs is the cancer cells themselves. And the way radiation acts is by leading to accumulation of nucleic acid in the cytoplasm, particularly DNA, which is activating a canonical pathway of defense against viruses, because that's what the viruses does, mediated by CGAS, the cytosolic DNA sensor that then produces SIGAM. SIGAM activates STING, and STING activates type 1 interferon production. And we stumble on this trying to actually understand why in our preclinical studies some radiation regimen were better than others at synergizing with the immune checkpoint inhibitors in inducing systemic responses, anti-tumor responses. And what we found that there was the radiation doses that were better, and hypofractionated doses of A-grade times 3 was in our models better than a single higher dose. And the difference was, after radiation in vivo, that there was upregulation of this set of genes that they all belong to the type 1 interferon pathway. And in fact, we did find that, a contribution that was important came from the cancer cell themselves by irradiating the cancer cell in vitro, and showing that this dose of radiation really increased the production of interferon beta by the cancer cells, similarly to what a viral infection can do. And this kind of summarizes a lot of work showing that indeed the production type 1 interferon led to increased recruitment to the tumor of these conventional dendritic cells type 1. They were also more activated, expressing CD4, a very important marker, and we'll come back to this in the end of the talk. And then when we, look in the brain and lymph nodes, there is activation of anti-tumor T cells, CD8 T cells, specific for an epitope that is a tumor-specific antigen, measured here by measuring gamma interferon production. And this occurs only when there is radiation combined with the checkpoint inhibitors, in this case, anti-CTLA-4. Radiation alone doesn't actually make it. It provides all this stimulation. It does by itself increase dendritic cells, but it doesn't overcome all of the immune suppressive barriers, so you need CTLA-4 in top. But then when you have the two of them, now you can find CD8 T cells that are actually not only going to the irradiated tumor, but also to abscopal tumors, so tumor outside of the radiation field. And this is an example of how this works. In a mouse model, this is a TSA tumor, is a mammary breast cancer, as a mammary mouse tumor. And there are, we use this system where there are two tumors. One is irradiated, the other is outside the radiation field. And this tumor is completely resistant to anti-CTLA-4. So there is really no response in either of the two tumors with anti-CTLA-4 alone. Radiation does cause a tumor growth delay, but only of the irradiated tumor. So itself is not able to elicit systemic responses that would be able to lead to scopal responses. But in combination with CTLA-4, we got a very nice improvement of the control of the irradiated tumor as well as the non-irradiated tumor. And this response was completely dependent on CD8 T cells, because in a mice you can deplete the subset of T cells and show that the effect is completely lost. So now the important question is, of course, whether in mice you can see a lot of things that don't occur in patients. And so this was a very important patient. You probably have seen my colleague Silvia Formenti presenting this story many times. But the reason I like to introduce it is because it is one of those cases where you understand that something is at least possible. You still don't know exactly how to make it more reproducible, but it tells you that it is possible. And the story of this patient, this was a non-small cell lung cancer patient who came to Sylvia's attention, having failed all possible effective lines of chemotherapy and metastatic disease. And this was before there was a checkpoint inhibitors approved for lung cancer. And Ipilimumab had been approved for metastatic melanoma, but not for lung cancer because it was indeed not effective by itself in this disease. And so Sylvia decided to actually try, after discussing with the patient under a compassionate exemption, to treat the patient with one of the many liver metastases with a hypofractionated regimen of radiation that was effective in the mice to induce a scopal responses in combination with the anti-CTLA-4. And they gave Ipilimumab to this patient in 4 cycle as approved for melanoma. And the patient, a few months later, actually became a complete responder. Now, there was no immunomonitoring, but we had a supraclavicular lymph node that was removed while it was responding because there was still signal at PET-CT. I looked at the node and stained for CD8 T cells. There was still tumor, but there were a lot of infiltrating CD8 T cells, suggesting that indeed this patient was rejecting his tumor. And he remains to this day alive and well without tumor coming back. So this is an amazing case and inspired the trial that now was published 3 years ago, where we actually tested the radiation with Ipilimumab using 2 hypofractionated regimens, C3x5 on 9.8x3, and Ipilimumab starting on the first day of radiation. I'm not going to go into details on this, but just briefly, the reason this was an important study for us is because there were on intent to treat 18% of patients with objective responses, systemic responses, so abscopal responses by RECIST criteria. And among the patients that completed the treatment, actually a third had objective responses. I just want to emphasize there was no real increased toxicity of the combination, but many of the patients were progressing very rapidly and did not complete the treatment just because they had very advanced disease. But it still gave us a great opportunity to try to understand what are kind of the mechanism of response. And one thing that was, emerged is by measuring in the blood type 1 interferon, there was a significant increase in patients with objective responses. This was, at day 22. So within 2 weeks after completing radiation. And it was not seen in patients that had progressive disease, including the patient that did not complete treatment, so were not kind of evaluable, but it was clearly associated with response, which suggests that the mechanism that we see in preclinical model is likely to play a role in patients as well. Now, we also tried to understand whether there was evidence of having induced T-cell responses against the tumor. And so what we found is that the patient with objective responses had an increase in the sort of expanded clone by using TCR repertoire analysis. We could detect expanded clone in the blood of the patients. And some, that suggested that they really represent tumor-specific T-cell clones. But this we were actually able to confirm in one patient, a complete responder by, where we had all the necessary material to actually sequence this tumor and predict neoantigen and screen the predicted neoantigens for recognition by T cells. And what we found is that there were 2 clones, one that pre-existed in the tumor but was not detectable in the blood and expanded early on and remained elevated throughout the course of treatment when the patient was rejecting his tumor. And the other was completely new, but he emerged with a similar sort of kinetics in his peripheral blood. And they both recognized a mutation in this gene, KPNA2. This is a nuclear importin. which was interesting to us. And we wondered, what is this gene and whether radiation plays a role in exposing actually this neoepitope. And there was prior evidence from Jack Nefji lab years ago showing that radiation indeed can induce the presentation on the surface MHC class I antigen of some peptides derived from genes involved in the DNA damage response, such as DNA repair enzymes. So we looked at this KPNA2. We couldn't test directly the patient that was responding. He had no residual tumor, and we did not have, a tumor biopsy from him that could be tested for this. But we used a similar non-small cell lung cancer in PDX mice and treated the tumor with radiation and shown that actually this gene is indeed upregulated by radiation. And this led us to think the hypothesis that, the transcriptional response induced by radiation can actually expose immunogenic mutations that are silent because they are present in the DNA, but they are not transcribed and translated into protein at sufficient level for this peptide to actually be seen by the immune system. And this is, the MHC class I antigen presentation pathway. After the protein is synthesized, it has to be digested in a proteasome. Then the peptides are transported by TAP into the ER, where they are bound to MHC class I, and finally goes to the surface where it can interact with T cells. So to address this hypothesis, we went back to the mice. And we use the 41 model because it's a, it's a model we are very familiar with. It was the first model where we actually showed that there was a increased response, that basically radiation of the primary tumor could induce responses, systemic responses to anti-CTLA-4 antibody. And it's rapidly metastatic to the lung of the mice. And so again, the mice don't respond to CTLA-4 alone, but there is an increased survival when you have radiation of the primary tumor in combination with anti-CTLA-4, will reflect the ability of the mice to control the lung metastasis, and this response we have, previously shown is mediated by CD8 T cells. And a good thing about this tumor that he has kind of not super high, but a close to 300 mutation. And so what Claire Lhuillier, who was the postdoc who led this work, she selected, potential immunogenic mutation that were expressed in genes upregulated in expression by radiation, and was able to demonstrate by vaccinating mice that 3 of them were actually immunogenic. 2 are presented by MHC class I, and the other by MHC class II, actually, of the mouse. She then eluted the peptides from the surface MHC class I, MHC class II of the irradiated or non-irradiated cancer cells, and it did confirm the upregulation. So we use AH1, which is actually a CD8 neoepitope that is not, is expressed by this tumor, but is not affected by radiation. And in fact, we don't, it's our control. It's expressed at high level at baseline, but not really increased by radiation. In contrast, this neoepitope expressed by CAN1, a gene upregulated in expression by radiation, is expressed at very low level, but markedly increased by radiation. We couldn't detect the other class I epitope, which, in either condition, suggesting that it's expressed at levels that are too low to be detectable with this technology. But we were able to actually see the class II neoantigen be presented by the cancer cells. And it's interesting because the expression of class II is very low on these cells at baseline, barely detectable, but it is increased by radiation. And actually this, leads to increased presentation also of this neoantigen. So they scanned one specific CD8 T cells. Well, actually, we confirmed they were only recognizing the mutated on the wild type peptide, and this recognition was blocked by blocking MHC class 1, confirming the specificity. And then Claire did take the 41 cells in a sort of competition assay. They were labeled with CSFE and incubated with the either non-specific CD8 T cells or CD8 T cells specific for the CAN1 neoantigen. When incubated with non-specific T cells, there is really no difference in the relative percentage of irradiated, non-irradiated cells. But when incubated with the CAN1 specific T cells, there is a selective reduction in the irradiated cancer cells. And so she showed that actually the irradiated cancer cells were preferentially killed by the neoantigen specific CD8 T cells. So vaccination of mice with this combination of neoantigens did not protect actually mice for development of 41 tumor, but improved the control of the irradiated tumor. And interesting also improved control of the irradiated, non-irradiated metastasis in the lung. And this was actually associated with some evidence of antigenic spread. So we can see an increased response against AH1, non-neoantigen, but a shared antigen that is actually not, present in the vaccine. We found the increase of T cells specific to CAN1 in the tumor. That was, the highest percentage was present in mice that were vaccinated and received radiation, but there was also an increase in mice that only received radiation, suggesting that actually radiation itself can prime some of these neoantigen specific T cells. But what radiation cannot do is to make them capable of producing gamma interferon measured here in the draining lymph nodes. Only the vaccine and the best response again is with the combination of radiation and vaccination. You see this really strong gamma interferon production in response to the neoantigen, both CAN1 and the other one. In the class 2 epitope, neoantigen really elicit very strong gamma interferon production by CD4 T cells. Now we know that both CD4 and CD8 T cells are required for the therapeutic response, because if they are depleted from the mice, we lose it. And also if we vaccinate the mice only with the 2 class 1 epitope, we don't really get this response. Now, to summarize here, a sort of additional mechanistic data in the list of time. I think there is somebody talking in the background. I don't know if maybe somebody has to silence their microphone. So to summarize here additional mechanistic data, we found actually that the CD4 T cells were not only important for fostering the helping the development of the CD8 T cells against the tumor and helping antigenic spread, but they actually were capable of directing also killing the cancer cells. And this was kind of helped by the radiation-induced upregulation of this ligand FAS and DR5. So in summary, really, vaccination with this radiation-induced neoantigen seems to be able to improve the control of the irradiated tumor, non-irradiated lung metastasis, and radiation is able to expose this otherwise silent immunogenic mutation that pre-exist in the DNA of the cancer cells. I want to now move to discussing limitation, focusing as an example on the combination of radiation and CTLA-4 blockade, which is where we have the most, we have done most of the work. And the key question here was particularly, after sort of being able to see that some patients develop anti-tumor CD8 responses after treatment with this combination. We wanted to really dissect what is the exact contribution of radiation and CTLA-4 blockade to development of these T-cell responses. And so Niels Lundqvist, and another very talented postdoc who now has his own lab at MD Anderson, devise a system where we can longitudinally follow up the mice. So they are implanted with 2 tumors. And again, we went back to our 41 model for this work. So one tumor is analyzed before start of treatment, and the other is analyzed after completion of the treatment. And he did both analysis of the TCR repertoire as well as RNA-seq of whole tumor. And this is just the take-home message here. When you look at mice not treated, the 2 tumors, you know, there is really no change in the infiltration by T cells. CTLA-4 alone also does very little, which is consistent with the fact that it has no therapeutic effect in terms of there is no real detectable increase in T cells. But actually, when we look at the TCR repertoire of the few T cells that are present, there is an increase in clonality. But radiation is really the one that drives increased clonality of the T cells in the tumor, and also a marked increase in T cell infiltration of the tumor, even by itself. But this is much more pronounced when you have the combination between radiation and CTLA-4. So we were curious whether radiation, you know, the contribution of the combination comes mostly from a quantitative or a qualitative difference. And to address this question, we dig deeper into the T cell compartment present in the tumor in the different condition by doing single cell analysis. And this included, you know, actually decamer specific for this common antigen in this tumor model, as well as TCR sequencing, so that we can put all these parameters together. And I just, you know, this work is not yet published, and I just want to give you some key sort of findings. This is a UMAP that, you know, showed we identified actually 17 clusters, and I want to focus on 4 of them. So this is cluster 1, which is a regulatory T cell cluster. So a CD4 cluster of regulatory T cells, which are cells that actually hinder tumor rejection. Cluster 0 is a cluster of CD8 T cells that actually express a lot of marker of cytotoxicity, as well as terminal differentiation. And they also express like FLI and PD-1, which are of course, you know, suggestive that they have a sort of exhausted phenotype. Now, cluster 1 was, you know, not really affected by CTLA-4 alone, but it was decreasing relative amount in the mice treated with the combination only. Cluster 0 was actually kind of decreased in mice treated with CTLA-4 alone in relative amount, but also in the mice with the combination. Now, but this was really sort of occurring at the because of the expansion of 2 other clusters. One is cluster 2, that is a CD4 T cell cluster with a helper polyfunctional phenotype. These cells express CD40 ligand, gamma interferon, TNF, and other chemokine and cytokine. So they are defined as polyfunctional, and they are really driven by CTLA-4 blockade. And they are present in sort of represented in both the CTLA-4, you know, in the cluster, in the mice treated with CTLA-4 or with combination CTLA-4 radiation. And then there is this cluster, which is particularly interesting, cluster 4, because this is a CD8 T cell cluster that is also driven by CTLA-4, but is really mostly expanded when you have the combination. And this is a CD8 T cell cluster that is also polyfunctional, doesn't really have markers of cytotoxic activity, but it produces as marker of tissue residency, and cytokines like gamma interferon, TNF, and other chemokines. And this is a marker actually of sort of survival resistance to apoptosis in this setting. And so it seems that when you have the combination of radiation CTLA-4, 3 key clusters of T cells, a cluster of cytotoxic T cells, a 2 cluster of cytokine polyfunctional producer CD4 and CD8 T cells now have equal representation. And so this is, of course, in this type of analysis, we look at relative proportion. We analyze the same number of cells in this condition. So we went back to the RNA-seq bulk data, and in fact, we found that all 3 of these clusters are markedly increased in the tumors treated with a combination of radiation and CTLA-4. And so what Niels did, he devised a signature that was cluster specific, and then went to check in actually patients using the metabolic database for triple negative breast cancer to see which patient had this signature. And what he found actually that the enrichment for the signature associated with cluster 0, 2, and 4 was increased in patients, was associated with the improved survival in the patients. And this was also in a multi-factor analysis, and it was not really reflecting just T cell infiltration, because there is really no association between CD3 as a marker just for T cells and outcome in this patient. And this suggests that these functional subsets of T cells are actually important in patients as well. Another question we asked is whether the antigen-specific CD8 T cells, which we could actually identify because we had this decamer, are sort of, well, are they found in the different treatment groups? And in untreated mice, CTLA-4 treated mice and radiation treated mice, the majority of CD8 T cells specific for the tumor antigen are all in cluster 0, but they redistribute between cluster 4 and cluster 0 in a mice treated with the combination of radiation and CTLA-4. So finally, all of this is very interesting, but we asked, radiation CTLA-4 in blockade in this tumor model don't really cure the mice. This is a waterfall plot showing that there is some mice that have a marked tumor regression, but we really don't cure them. And so we thought, well, can we add the PG1 or LAG3 blocking antibody and improve the responses? They were really found in this cluster 0 of effector T cells. And to our surprise, the answer was no. PG1 addition did not help. LAG3 targeting antibody didn't help. We also tried other antibodies that target regulatory, molecules expressed in the tumor in the regulatory T cell compartment, and even in combination, there was really no improvement. But the improvement was seen markedly with blocking, actually with agonist CD40 antibody that target the antigen-present T cell compartment, so the myeloid compartment. And we did sort of see the, we confirmed that it was really the 3 treatment, radiation, CTLA-4 blockade, and CD40 were all required to achieve this result, because when you only use 2 of them, radiation, CD40 alone, or the 2 antibodies, CTLA-4 blockade, CD40 alone, they don't really do the trick. So you need both. And we are still completing this work, but I think, kind of the take-home message is that CTLA-4 blockade really seems to promote polyfunctional T cell differentiation, while radiation diversifies and expands the TCR repertoire of T cells. So new priming of T cells is a very important contribution of radiation. When you use them together, now you have both effect, and importantly, you seem to have a major shift in the CD8 compartment from a cytotoxic function towards a cytokine producer. And, you know, I think it suggests that you really need all these different functional subsets to control the tumor. And finally, you know, I think it's important to consider, you know, very often a lot of trials have been done. If one checkpoint doesn't work, maybe two will, because of course there is some evidence that they can improve responses, but this may not always be the case. And maybe we have to think that, you know, even if PD-1 is expressed, it doesn't always mean that is going to be the best treatment. And maybe it's important to consider targeting different compartments in the tumor, particularly, you know, in some tumor types, the myeloid compartment may be quite important. I want to go quickly over the barriers. Before I go into the adenosine, I just want to go back to the concept that interferon type 1 is important, and it is produced following radiation, you know, because of the DNA in micronuclei goes, as I mentioned earlier, and stimulates CGAS. This leads to type 1 interferon activation. Now, we know that, you know, there is multiple regulatory mechanisms. One is the exonuclease CREPS1 that can actually cleave the cytosolic DNA and kind of blunt this response. Another important mechanism that was shown last year by my colleague Lorenzo Galluzzi is autophagy, that actually kind of removes damaged mitochondria that contribute an important source of the cytosolic DNA that activates CGAS and STING. There is, in addition, the epigenetic downregulation of CGAS and STING themselves that was nicely shown in this work. And so many tumors lack or downregulated expression of these important molecules. And here I'm just showing an example from our trial of a lung cancer patient that actually really seemed to not express STING in his tumor, while, and this was a non-responder. One of our responders did express it, but we didn't have enough data to really make any conclusion that this was an important mechanism. But it might be something to keep in mind. So I just want to cite two studies, one, you know, related to TREX1. This is a very interesting study done by the Chicago group of Steven Chumland and colleagues. And what they did treat patients with multi-site radiation with pembrolizumab. And they had, you know, responses in a radiated tumor associated with survival. Interesting, looked like pembrolizumab helped the response to radiation of the irradiated tumors, because large tumors that only receive partial radiation were responding as good as tumor completely irradiated. But the most important reason to look at this trial is because they were able to do in some patients paired biopsy before and after radiation and show a regulation of actually of immune pathway in the biopsy post-radiation. And they also looked at TREX1, and it's interesting that they found that upregulation of TREX1 was associated actually with progression of the lesion in this small set of patients. It was about a dozen of patients, but nevertheless, it's an intriguing evidence of the potential importance of this mechanism. Coming back to the epigenetic downregulation of STING, work by David Balby's lab has shown that some subtype of lung cancer defined by LKB1 mutations actually have epigenetic downregulation of STING. They are very resistant to checkpoint blockade and you know, are insensitive to cytoplasmic DNA double-strand sensing. And so, although there is really no data that I know of about this tumor being or not able to respond to radiation by sort of, you know, if radiation could help improving the responses of this tumor, but it is interesting to consider whether actually, you know, we could look at some of these tumor subtype to find, to sort of hypothesize, you know, and ask the question, are these the tumor that may not benefit from radiation? I think this is important in tailoring this type of approach to the patients that may benefit the most. And finally, what we asked is whether there are alternative pathways to actually recover the ability of radiation to really increase the infiltration of the tumor by dendritic cells type 1. That's where I started. They are important to actually make the tumor responsive to immunotherapy. And what we focused on was the sort of ATP and adenosine pathway, because this is a very important pathway in regulating anti-tumor immune responses and inflammation. And in the tumor, there is a lot of release, particularly after radiation of ATP. In fact, ATP is one of the critical markers of immunogenic cell death, because ATP itself is very inflammatory when found in the extracellular compartment, and it can activate dendritic cells as well as other immune cells. But it is very rapidly converted by CD39, CD73 into adenosine, which has the opposite effect, is anti-inflammatory immunosuppressive. There is also release of NAD+, which is catabolized through the non-conventional or alternative pathway, non-canonical pathway by CD38 and CD203A into, again, the AMP, which then CD73 converts into adenosine. So Eric Wennerberg, another really talented postdoc who now is a junior leader at ICR here in London, a junior team leader at ICR in London, led this work. And what he found is that radiation upregulated the expression of the ectonucleotidases involved in the non-canonical adenosine generation pathway, CD73, CD38, and CD203A in a dose-dependent way. These are two different mouse breast cancer cells, and he also found the same in human breast cancer cells. And in the tumors, he found actually increase in adenosine, which was also radiation dose dependent. So since CD73 is the common enzyme for both pathway, he blocked CD73 and in fact found that it could improve the response of the tumor to radiation and the survival of the mice. In this case, used with a 20 gray single dose, which was where there was the strongest accumulation of adenosine. He also confirmed that the expression in the tumor was important by knocking down this gene in an inducible way in a TSA cell. And I just very briefly want to point out that, adding when you knock down CD73 from the cancer cells, blocking also with the antibody doesn't seem to add additional benefits. So a large contribution, at least in this model, comes from the expression of CD73 in the cancer cells, which is upregulated by radiation. Now, how was blocking CD73 improving tumor responses? He found an increase in dendritic cells type 1 in the tumor. And in fact, mice that lack these dendritic cells completely, did not respond to the combination, no improvement with the combination, indicating that they are critical. And they went on to actually develop dendritic cells, CDC1 in vitro, label them and introduce them in BACTA-free deficient mice that lack these cells, and then measure whether they were homing to the irradiated tumor. And in fact, they found that they were homing only to the irradiated, but not to the non-irradiated tumor, but only when CD73 was actually blocked. Suggesting indeed that blocking adenosine is a critical alternative way to get dendritic cells into irradiated tumor. And finally, he did a very important experiment. If you have a tumor that is deficient in CGAS, and we know this tumor don't really recruit dendritic cells well after radiation, with a grade times 3, because they don't make good Taiwan interferon. Can you restore this recruitment by blocking CD73? And the answer was actually yes. Now you have a nice increase in the dendritic cells homing to the tumor. And most importantly, also was able to restore scopal responses and improve the survival of the mice when CGAS is deficient in the irradiated tumor in combination with the anti-CTLA-4. So I'm showing here the survival of the mice that reflects control of both tumors. So when you have a CGAS-deficient tumor and you treat with a great times 3 and anti-CTLA-4, there is improved survival. When you have CGAS-deficient tumor, now you do not have this effect, but you can recover it by adding CD73. And now, to mention, however, the soluble, the anti-CD73 alone doesn't really help a scopal response. It helps control of the irradiated tumor, but not a scopal responses. And it's kind of a really interesting sort of additional piece of information, how combining different modalities is really important, depending on what is the goal you want to achieve. And I just want to conclude here by kind of summarizing the sort of, what I presented to you today. So the recruitment of dendritic cells type 1 is really critical for the ability of radiation to synergize with checkpoint inhibitors, and is promoted by at least 2 pathways, interferon type 1, but also the ATP and AD pathway seems to be important. Interferon is, dependent on the expression by the cancer cells of STIGAS and STING, but is regulated also by multiple other processes, including TREX1 and autophagy. And the ectonucleotidases that convert ATP and NAD in immunosuppressive adenosine also regulate this alternative pathway, CDC1 recruitment to the tumor. And so I think that it is important to consider to improve the success of combination of radiation and immunotherapy, particularly immune checkpoint inhibitors. It was going to be important to understand the characteristic of each individual patient's tumor and then design a combination treatment that may address barriers and missing sort of points, missing mechanism. And I just want to mention the people that, did the work. Maud, who is still in the lab and is finishing up the story with CD40. Niels, Claire, Louillier, and Eric Wennerberg, who all now have their own jobs, and I'm very proud of them. And this, of course, my long-term colleague and collaborator and friend, Silvia Formenti. And this is our program at, growing program at Weill Cornell Medicine on radiation immunotherapy and other collaborators in our funding. And thank you very much for your attention. And I'll stop sharing now, and I think.
Thank you so much, Professor Demaria, for the excellent talk. So I think we have a few questions already in the chat. Crispin asked the first question. Are you still on the line? Do you want to ask the question yourself, Crispin?
Hi, Sandra.
Hi.
Thanks.
Hello.
Wait, so just a quick question, just because can you hear me?
It's freezing. Maybe I can read it. Given the importance of butterfly dendritic cells highlighted in the first part of the talk, what happens to the frequency of tumor residing butterfly disease following radiation? So we actually want to look at that. We did only analyze in single cell analysis. We analyze only T cells. We saw T cells from the tumor. So we do not have the data because, of course, as you know, the myeloid compartment is very complex, and there are multiple emerging, state and subset of antigen presenting cells. So it is a really important question, but we haven't done it yet.
Okay, thank you.
Sure.
So second question from K81819554.
Yeah, it's me, Azad. So
please.
Hi.
In short, that in one of your cluster, you said that in cluster 0, CD8 T cell is less in combination treatment, whereas in cluster 4, CD8 T cell is increased in the combination treatment, RT and CTLA-4. Any explanation for that or any reason?
Yeah. So it is a relative increase, of course. When we look at the bulk RNA-seq data, in total number, they both are more in the tumor. There is a lot more T cells of both types, but their proportion are changing. So in untreated tumor, there is, and in tumor treated with only radiation, there is a huge dominance of cluster 0, which are these, kind of exhausted, terminally differentiated effector killer T cells. And when you have the combination radiation CTLA-4, you drive really these different subsets of CD8 T cells that are a cytokine producer.
I see, understand.
Yeah, so I think, because they share the same antigen specificity, when we look at that part, they obviously, I don't know if, these are somewhat newly primed cells that differentiate differently from the beginning of they shift their state. As you know, John Werry has a lot of beautiful data on how epigenetic shifting of controls T cell states after activation. So we don't actually know the details of that.
Anthony, just a quick question. Just sorry again. I probably missed it, but in one of the earlier slides, in first part, you showed that RT-CTLA-4 reduced tumor growth in normal tumor as well as in the abscopal effect. Does it is also similarly effective in abscopal effect as well on the other tumor?
Well, yes, it is, but I should say there is some model dependency in that.
Oh, I see. Yeah.
In the TSA tumor, we get really good responses, but if the tumor are very large, there is an effect, but it's rather difficult to get the tumor to go away completely.
Yeah.
So I should say that there is probably scopal responses. They are amazing when you see them in patients.
Yeah.
But there are many barriers to get them, and it's definitely not an easy one, even in mice.
In mice,
yeah.
Yeah. Thank you.
Sure.
Next one is Swati Panghaka.
Yeah, thank you. Great talk, Professor Demaria. I just wanted to ask if you looked at the myeloid cell populations within your radiotherapy and anti-CTLA-4 treated tumors, and whether the macrophages polarize from immunosuppressive phenotype to an inflammatory at all in the responders specifically.
Yeah, that's a very good question. We have looked more at the dendritic cells, not so much at the macrophages, to be honest. We have looked more in the setting of, the mice treated now with the CD40, of course, where we do see a change, but it seems to be really driven by CD40 agonist and not occurring with radiation alone, at least in this tumor model. And, again, I think we have to be careful not to maybe generalize everything, but I think there are a few very important things when it comes to the, macrophage compartment. I think one is a kinetic, because, a study by other investigators have shown that there may be some changes that occur over time, and if you wait long enough, you are more likely, unless you develop a good immune response, you are more likely to recruit from the circulation macrophages that then monocytes, rather the cause of that then differentiate into M2-like macrophages, repair-like, so immunosuppressive. But if you do achieve, if you now have T cells coming in, you do get gamma interferon produced, and now you get the new monocyte that come in to differentiate more towards an M1 and antigen presenting cell or even cytocidal macrophages. So I think it's it's difficult to make a conclusion, but I think radiation alone, when you have a tumor that is very non-immunogenic, it's not a good at shifting the macrophage compartment sufficiently to an M1 or, or a anti-tumor phenotype.
Thank you.
Okay. So John Anderson next, please.
Thank you so much. Really great and very interesting. I'm particularly interested in, what you might call ultra cold tumors which are cold because they're, highly devoid of neoantigens. And here, and particular childhood tumors, which I work. And here, we use radiation as a standard of therapy. So I'm particularly interested in whether there's a role for, gas sting in recruitment of innate effectors, such as NK type cells, which, potentially very important where there is no naturally occurring adaptive immunity.
Yeah, so that's a very important question, because when we work with mice model, it's, there is some degree of immunogenicity, maybe artifactual in many cases. I don't know. The answer is that, I haven't worked with really good model of pediatric tumor. What I know from the literature, and one way radiation could help is, and we are actually sort of seen to some degree in some of our model, it's work that is very early, so I haven't spoken about it yet, but there is some epigenetic changes. So I think that the potential of radiation to induce antigenicity in pediatric tumor may come from maybe reactivation of either endogenous retroviruses that can also stimulate interferon responses, through the other pathway, right? The RIG-MAVS pathway, or and potentially also encode interesting antigen. One thing that we are working on right now also is other types of antigen that radiation can induce, which are not genomically encoded, not genomically templated, but post-translational modification of different type. I think there is a very big, a lot of work is being done in general, how important some of these crazy antigens, basically non-canonical proteins, other, like, when you skip the tryptophan, Yardena Samuel said a beautiful work on that. And I think there was a very nice work by a group in Paris on pediatric tumors showing actually that there is some subset of them that are immunogenic, and they seem to be immunogenic because of this epigenetic dysregulation. So I think that's a very, very interesting place to look at, and very important actually. I think is a fascinating and also very, of course, a big area of need. So I just, I don't have much information myself, but I actually thought a bit about that. I think is a very nice, very important place to look at. I would bet that you find something by looking at the RNA pathways and the, and possibly interfere through that pathway.
Thank you. Next, Rahul.
Yeah, hi. I wanted to ask you whether you think that your research has been able to prove that radiotherapy has an unequivocal role in converting cold into hot tumors. And I also wanted to ask you what is your take on combining radiotherapy with more conventional agents, such as targeted therapy or emerging therapies, such as CAR T-cell therapies in solid tumors?
Yes, so what, I'm perfectly, humbled that we cannot declare that we can convert cold tumor into hot tumor with radiation. I think we can help. Now, one, issue to keep in mind is what do you define as a cold tumor? And very different definition have been used. I think there are tumors that really have very few immune cells whatsoever, others that have a lot of immunosuppressive myeloid cells, which I think is kind of more likely to be the case, for instance, in breast cancer. So those are probably presenting two different challenges of how you can, convert those tumors into hotter or at least lukewarm and potentially hot tumor. I think that, it is challenging to find the perfect model in mice to ask that question. But in terms of, if you have a really very tumor that is really excluding completely immune cells, can you change the phenotype? And I have to admit, we have not tested such a model where we can be absolutely sure that that's going to be the case. That said, I must say that, what we know about tumors, that at least in adults, they all have evolved. I mean, there is more and more evidence that all tumors have evolved by escaping the immune system. And so, some memory towards the tumor may actually be present even when the tumor seems to be cold. So, a really key issue to address at the conceptual level. And then at the practical level, I think what you want to know is, are you going to treat the patients, right? And which patients are likely to respond. And in that, I think is a little too simplistic to label cold or hot. Particularly because that's often done just based on PD-L1 expression, which is really not a good sort of measure for that. I think, we have maybe to go beyond the cold and hot and really understand a little more about the phenotype of the immune cells present in the tumor and what are the immune barrier. If you have a very active adenosine generation, either expressed by the tumor or by the tumor microenvironment, it's unlikely maybe that you will succeed without having an agent that blocks that pathway. If you have, blocks into the interferon type 1 pathway activation, then you have to think of maybe adding agents that stimulate type 1 interferon, like TLR agonist, potentially STING agonist, and so on. So I think it's really, basically, I think we cannot think of radiation being added to every patient that fails immunotherapy and being able to do something good. But we also should not discount what we need to know is which patient can benefit. There are clearly patients that can benefit. And it's just that we aren't good at identifying them and identifying what combination is going to do the trick. And, and so I think that's where real progress is going to be made by really studying better where radiation can benefit and how to use radiation to get the most benefit.
Thank you. Next is Farah.
Hi, thank you so much for the great talk. I just had a question with regards to the fact that you've kind of shown the changes in the 0 and 4 clusters for the CD8s in the work that you've kind of shown for the 421 model. And so please correct me if this is wrong, but you've shown this in the context of the 3x8 gray standard therapy of radiotherapy that you've used. I just wondered if you've also looked in the context of a single dose, so for example, like a high dose of 20 grays, and if that is the case, kind of have you seen then any observation or changes within the clustering of the CD8s as well?
Yes, no, I think it's an important question and we haven't. That's a very simple answer. We did not test 20 gray. Part of the reason is we do not really see therapeutically a good synergy or additive effect, whatever, but a big good therapeutic effect in the model that we have tested with 20 Gray and CTLA-4 inhibition. That said, clearly there could be changes that actually are very important and that could kind of indicate maybe what is happening. So I think it's an important experiment to do. It's just that it's a rather expensive experiment to perform, and so we haven't been able to do all that we would like to do, but I think it's an important one.
Sandra, thank you.
Yeah.
And just want to ask a question. In terms of mutational neoantigen, you showed the effect of radiotherapy. Have you seen the difference in the radiotherapy sensitive versus radiotherapy resistant tumor? Is there a difference between different tumors if they are primary radiotherapy resistant, or if they're sensitive first, then become resistant, versus those primary sensitive?
So we really haven't looked, I think, in enough tumor to have an answer to that. But to sort of the ability of radiation, of course, in terms of, I think you do need to, there are a few things. So some radiation has to be able to do a few things to, I guess, properly expose pre-existing immunogenic mutation, which include sort of upregulating genes that, so you have to have a certain mutational burden to start with, because if you only have a couple of mutations, you are not winning the lottery of, you know, that they have to be in genes that necessarily are going to be sufficiently regulated in expression by radiation, or that those mutations are the right one to be seen by the immune system. So the number game help very much, as of course we know from the fact that tumors with higher mutational burden are more likely to respond to therapy. I think where this effect of radiation is useful is when you have tumors that have a fairly good mutation burden by many predicted potential neoantigen, but you know they don't seem to respond. And then the question is, can radiation now, you know, by increasing their ability to present, to make more visible those antigens, can that occur? How do we sort of, in terms of radiation sensitivity, to me, it is a bit of a difficult question, because I guess one, in part because I am not a radiation biologist, but obviously you know, if you think of radiation sensitivity in vivo, I think it is kind of affected by, you know, in immunocompetent host, at least it's going to be difficult to separate, right? How you trigger the immune system versus how you control the tumor. And so basically what I want to say is that if you don't have T cells in the mice, if you don't have an adaptive immune system, then you are really looking at how radiation is able to control the tumor directly. But the same doses in mice with a T cell response have a different effect. And this was actually shown by Luca Milas a long time ago, you know, when he showed that the radiation that controls 50% of the tumors is, you know, it's a lot higher in mice without T cells and a lot lower in mice with T cells. So I think it's a difficult sort of the concept of radiation sensitivity, at least in my mind, not being a radiation oncologist, is a difficult concept to establish, because to me it's like, we can use radiation that by itself does not control the tumor, but now when you add immunotherapy, it's a good dose to control the tumor, because you have that synergy. And you know, I'm sure I didn't really address your question properly, Anthony, but
you got a good point, Sandra. It is more complex, isn't it? So I think that we got one more question from Crispin. Obviously, we are over the time already. So Crispin, do you have the last question?
Yeah, Sandra, if you have time, I'm just going to leave my camera off just because in case it's my bandwidth.
Sure.
But and this might be a silly question, but so obviously you talked about radiation-inducing expression of neoantigens, and a long time ago in my PhD, I messed around with radiation-responsive promoters, and
Yes.
obviously the cell doesn't know whether the gene that's being expressed contains a neoantigen or not. I mean, some genes are the expression is being increased following radiation, and I guess the question is, do you have any understanding of, why certain genes might be increased following radiation or not?
Yes.
Is it all genes? Is it some genes? And if it's some genes, do you think we could ever sort of computationally work out, why that is? Is it because they share a promoter? And if so, you could imagine an idea where, I guess this is kind of speaking to Anthony's question, where you sequence a person's tumor, you can see where their mutations are. You can try and predict neoantigens, which is a notoriously difficult thing to do. But then you could look at what promoters control those genes that contain neoantigens, and whether you think that patient might have increased expression of those in response to radiation. I just wonder if that's something you'd thought about.
Absolutely, you are exactly sort of describing what we are actually working on. So the idea was, could you predict basically by having a tumor of a patient sequenced, the mutation predicted, and then say, oh, are these potential immunogenic mutation expressing genes likely to be upregulated by radiation? And is that a predictor, it becomes a biomarker predicting whether the patient could benefit by addition of radiation to checkpoint inhibitors? And so we are trying to address this question. So we have used, and I think that is a question that is really important to address in patients and not so much in mice, because, it's not really. So we have, we started in non-small cell lung cancer because, of course, it's a good tumor that has, most tumors there is a broad range, but many have a sufficient number of mutations to have at least a few that could be a good candidate. So we are using actually right now organoids to address that question, so that we can do the RNA-seq and then the elution of the immunopeptidome and confirm whether it's really happening. It is, as you may imagine, a very time-consuming work because you have to expand a fairly large amount of these cells, and they don't grow very fast. But we want to do a number. Right now we have a few, and we start putting together a list of genes that are commonly upregulated by radiation. Still, you can say, but of course, why genes are upregulated by radiation? So a few things. One is because, of course, there is a huge DNA damage response, and genes that are involved in a DNA damage response are often mutated in tumors, because that's part of the reason the tumor becomes, a tumor, because it has a dysregulation of those processes. So it looks like maybe it's a set of genes that tend to be enriched for mutation. In addition, I think, if the radiation upregulates the type 1 interferon pathway, actually is a very large number of genes that can be upregulated in expression. And one of the genes in 41 was actually a gene that is in the interferon type 1 pathway that was upregulated. I think other genes are really involved in a protein turnover, because that's also part of, when you have such a damage that is a lot of oxidative changes, affect a lot of biomolecule in the cells besides the DNA break, a lot of pathways are activated. But, the key here is there is also a lot of variability. We have done RNA-seq of a few different human lung cancer. In some cases, you have 1,000 of genes upregulated. In others, you have a couple of hundreds. So there is a big variability. And the key question is going to be, do we have a reliable call of genes so that we can expect to be always upregulated? And I think, indeed, is a place for computational biologists to help out. And, so if you have, you know, if you have an interest, it would be fun. Maybe we can get in touch.
Yeah. No, I'll email you. No, I think that's a really important thing to look at, definitely.
All right.
We've got a lot of lung cancer data, but not a lot of lung cancer data following radiation. But
right. Yeah.
Thank you very much. That was an excellent talk, and obviously don't normally get quite so many questions. So I think, you know, and people aren't normally here at, you know, half an hour afterwards. So that was really fantastic. Thank you very much.
I can see that everybody's so interested, Sandra. So I think we are 20 minutes over the time. So I think we'll probably conclude the seminar today. So we thank you so much for you to actually give us a talk, and also thank you for everybody who attends this seminar and for staying so long. To hear the questions and answers. So thank you, everybody.
My pleasure. Was fun, and hope to see some of you in person in the next future.